Yes, so as Konstantin already said, I consider myself a comparative neurobiologist or neurophysiologist, and we work with two animal species in my lab, which is the macaque monkey and the carrion crow, and I will show some data of these two animals. And in collaboration, we also are able to do single-cell recordings in human patients with the University Clinic in Bonn with epileptologists there. Today, I will focus on working memory. But before I do, this is the menu for today. And before I forget, you're welcome to interrupt me in case there's anything you would like to know in between. So I thought it would be a good idea to first define what I mean by working memory, or what the textbook means by working memory, because it's important to me that it's not mixed up with other types of short-term memory. Then I talk briefly about history, if you like, how neuronal correlates of working memory, and by neuronal correlates today I exclusively mean single cell correlates, have been discovered many decades back. Then I switch to my own work and talk about monkeys and how they memorize abstract categories, because numerosities, numbers, is one of our core topics. I will talk about memorizing numbers today a lot. And then also show you some data when monkeys are using numerical information in their working memory to guide their behavior according to switching rules. Then I switch over to crows, and here I will firstly introduce a little bit the differences between the cerebral cortex of mammals compared to the pallium, the telencephalon of birds and crows, because there are some very interesting differences that are important for me as biologist. And then basically introduce very similar neuronal correlates of working memory in the crow, nidopallium caudolaterale, which is the supposed equivalent of the prefrontal cortex. But I'll come to this, and then I try to wrap up everything by trying to put out ideas how people envision that these working memory signals are maintained in the brain. So when we talk about working memory, then we talk about a specific type of short-term memory. And in this display here, you see the standard processing line if we receive information from different sensory channels, seeing, hearing, and so on. This is first loaded into a sensory buffer that is only lasting for a few tens of seconds, and then after that it has to be loaded into a short-term memory workspace. Short-term basically usually means a few seconds, a few 10 seconds. In this short-term memory, there is one specific type of memory that is called working memory, and we also need this type of working memory to retrieve information that we have in long-term memory. If we want to consolidate what we have in our working memory, then we need to rehearse it. We need to try to put it into long-term memory. But as I said already, long-term memory is not consciously accessible. If we want to load this information from long-term memory, we need to retrieve it back into working memory. So what is working memory? It is a temporal storage of a few seconds, tens of seconds, but very important, it is more than a simple storage. It is the temporal space that allows us to manipulate information. So we process this information, we change it in our working memory space. Think about doing a mental calculation. This is my beloved example for working memory. You keep, so if I tell you 2 plus 3, you keep these numerical values in your working memory, but you are also manipulating it because you are adding them up and you come up with a solution that this is 5. This is a classic example of working memory. Working memory has specific characterizations. So first of all, it is a limited buffer. We cannot load an endless amount of items or features into working memory. Most people think that we can load up to 4 different features in our working memory. Some people even think it's just 1, and we temporarily multiplex this information so it looks like 4. But I think 4 is a good number, so the magical number 4 here. So this is limited capacity. Another core feature is that it is very distractible. We actively, by cognitive control, need to safeguard information that we have in our working memory. Again, an example, if someone tells you a telephone number and you go to the telephone, try to pick it up and try to punch in the numbers, and someone else would shout out a different number, you would have a very hard time to memorize this original telephone number because you actively need to suppress distracting information. And the final and certainly not least important aspect is that it is an implicit type of short-term memory. In contrast to many other types of short-term memory that are implicit, that are unconscious, working memory is always conscious. And I find this very interesting, and connecting this to the beautiful symposia we had over the years. So when an animal has proper working memory, this is an indication of a conscious process already in itself. So a few bits about history. How did people start to investigate working memory and what did they discover? One of my heroes is Joachim Fuster in California, University of Los Angeles. He did the first seminal recordings in monkeys that were trained on a very classic working memory task. And this classic task is called a delayed match to sample task that I will also use extensively throughout my talk. It's a very simple task, but it has a working memory component. And here you see, for instance, such a monkey facing a screen, and it is presented with the color red. And the monkey's job is simply to remember the color red over a brief delay period, which in this case is 15 milliseconds, which is quite a long working memory time. And after this delay time, the monkey is supposed to choose the correct color among two different possible colors, blue and red, so the monkey picks red. And in doing so, the monkey tells us that he could hold this information red in working memory. And during this task, Fuster and his people were recording from the prefrontal cortex. I will show this later. So this frontal part of the monkey's brain, the highest integration center that we think of in our brain, while the monkey was performing the task. And you see some data here. This is neuronal data depicted as dot raster histogram. I will use many of these depictions, so I briefly explain what you see here. Every line here is one trial, and every little bar or dot in the future is one action potential. So you see the impulses of a single neuron across many trials that have post hoc been sorted again. And yes, so x-axis is time, and the y-axis here is the number of different repetitions. And you see these two displays whenever the monkey was presented with the sample color red and the matching color red. Whereas in these trials here, same number of trials, the other color was presented, the color blue, and the monkey had to match blue to blue. So we have only two conditions, which keeps it very simple, but also very informative. What you can already appreciate when you look at these dot raster histograms is that in this delay period, when the monkey is memorizing the color, you see a much higher discharge of this neuron when the monkey is memorizing red compared to blue. And the same information is depicted here as a peristimulus time histogram, where it's basically a column graph where every column or every bin contains the summed up number of action potentials. And basically, whenever these colors go up, it means that the neuron on average fired more. The blue columns indicate the firing rate for the trials when the monkeys were presented with blue. And you see the neuron doesn't care about blue at all. It has the same firing rate in the spontaneous, in the fixation period, during the delay period and afterwards. But the red columns, they represent the action potentials whenever the monkey was memorizing 3. And here you see very nicely, the neuron is dramatically increasing its firing rate, specifically during the delay period. So it's increasing the firing rate, and it maintains this firing rate up until the end of this very long delay period. And this is what many of us, including myself, consider as one of the most important neuronal correlates for working memory. This type of sustained activity during a delay period when the stimulus has vanished already, but the new choice stimulus is not yet available and the monkey has to memorize this information. Very similar kinds of responses have been shown by Patricia Goldman-Rakish at Yale University for many decades. She was primarily interested not in what information, such as color information, but where information. So where is an object remembered? And she was famous for doing this oculomotor delayed response task, where the monkey is fixating on the screen. And while the monkey is fixating and the eye movements are measured, of course, a certain cue is presented on one of 8 different positions around the clock on this display. This cue is removed again, and for 2.5 seconds in this case, the monkey has to remember where the cue appeared. And after this 2.5 seconds, the fixation stimulus is removed, and this is the cue for the animal to give a response. And in this case, the monkey was supposed to make a saccade, an eye movement, to the location where it remembered the target. And these targets were presented, as I said, on 8 different locations around the clock. And what she found regularly was that the neurons cared a lot about the spatial memory. So here you see a neuron that is only increasing its firing rate during the delay period. That's this shaded time interval when the monkey memorized a target that was exactly to the left of the fixation target here at 90 degrees. And the neuron didn't care at all about all the other directions that the monkey was memorizing. So here we have a neuronal correlate of spatial working memory. One of the very important questions that popped up pretty soon was, of course, single cell recordings can give us a neuronal correlate for working memory, but it doesn't necessarily mean that these neurons are causally related to the monkey memorizing a specific item or location. So people wondered about the causality of these responses, and I'm now going back to one of my beloved studies from Joachim Fuster again. One of the ways to investigate causality is to inhibit or to suppress neuronal activity temporarily, ideally, in order to see whether you end up with behavioral deficits. The idea is if these neurons are relevant, causally related for working memory, if you turn them off for a while, the monkey should no longer be able to hold this information in working memory. And this is exactly what Joachim Fuster did. In this case, he was also using a delayed match to sample task, but this is one of the rare cases where he used a tactile delayed match to sample task. So the monkey had to feel with its hand the shape of a cube, for instance. He couldn't see the cube, could only feel it. Then came a delay period where the monkey had to memorize this shape, and then came this choice period where the monkey could choose between 2 objects, feel them tactilely, and then decide which one was the one he remembered. These here on the top right corner are the conditions when Fuster cooled down big parts of the prefrontal cortex. And cooling is one of the methods to transiently inhibit neurons. Cooling means that they are being inactivated. But the nice thing about cooling is that it's transient. So after a while, based on the blood flow, everything is warming up and the neurons are functioning as always. Now, when he was cooling the prefrontal cortex, he found a very important behavioral deficit. Here is the behavioral situation. We have the percent correct choices of the monkey plotted against different delays. Different delays are not very important for me. Percentage correct means how often the monkey was choosing the correct object in this case. And because we only have 2 objects, chance level is 50%. 50% would mean that the monkey is guessing. But what you can see here, and these the triangles are the control conditions where everything was warm, where everything was normal. And you see the monkey can nicely discriminate the 2 shapes. for a delay of 10 seconds at about 90% correct. The interesting aspect happens when the prefrontal cortex was cooled down to 15 degrees Celsius. And these are the data here, the circle data. You see that there is a significant drop in the performance of the monkey, and the entire function is shifted downwards. And this is indicating that the monkey really has memory problems after one part of the prefrontal cortex was cooled down. So this is clear evidence that the prefrontal cortex is important for the monkey to memorize what it had just felt. You may wonder why the monkey is not guessing or the performance is not dropping to 50%. Well, one reason is that the other hemisphere was still intact. And another reason is certainly that only parts of the prefrontal cortex have been inhibited, and other parts of the prefrontal cortex can take over. But the important information was there is really significant deficit. As a control, he was cooling down another high-level area in the posterior parietal cortex, the classical association area, that also shows delayed activity neurons. But when he's cooling down this part of the brain, the monkey shows no working memory deficit. So prefrontal cortex, one of the main messages of this talk, is extremely important, is a key area for all sorts of working memory related activities. Now I'm moving over to my own work. And at first, we are also using a delayed match to sample task because I really like this task. And instead of colors or spatial cues, we are using very abstract numerical category. We are using numerosity or number, if you like, that the monkeys have to encode and have to memorize and then choose later. Protocol again works like this. The monkey is sitting in its recording booth and is facing a computer monitor, and the fixation stimulus comes up, and the monkey is required to maintain fixation on the central fixation dot. That's necessary because the neurons in all of these areas we are recording from are A, sensitive to eye movements, and we don't want to measure eye movements, and B, we want to have a stable visual presentation field when the monkey is watching these images. After the fixation period, a sample stimulus comes up, cueing the animal for a certain number of dots. In this case, the monkey is seeing 4 dots, but we are varying the number of dots in different trials. The monkey has to memorize the number it has just seen over a brief delay period of 1 second. And after this delay period, a test stimulus comes up, which in 50% of the cases shows the same number of dots, but importantly in a different layout. And the monkey is then required to respond, to tell us that it has now seen twice in a row the same numerosity. And it's doing this by releasing a manipulandum, a bar that it has grabbed at the beginning of the trial. In the other 50%, we are showing a so-called non-match. We are showing fewer or more dots on the screen. And in this case, the monkey is required not to respond, but to hold the bar until the second test stimulus appears, which is always a match, and then respond. And here again, we have a chance level of 50%. Just for those who are interested in number encoding, we do everything to make sure that the monkeys are not encoding the visual pattern or any physical property of the dots or the numbers of dots that we are showing. We are presenting the dots on random locations on the screen. We have control protocols where the area of blackness is kept constant, control protocols where the total circumference of all the dots is kept constant, where the density of the dots is constant. We vary from circles to other geometric figures. We present these dots in a linear arrangement or in a two-dimensional arrangement. And just to cut a long story short, whenever we control these factors and the monkeys are highly trained, they do not care about these visual properties. All they care about is the number that they have to encode and to memorize. When the monkeys are highly trained, we go in and record typically from 2 areas simultaneously. What you see here is a lateral view of a macaque monkey brain, very similar to our brain, just not as heavily folded. This image here shows you the classical visual pathway from the eyes via the lateral geniculate nucleus to the primary visual cortex, and from there over the dorsal path to the posterior parietal cortex, which is not only a visual area, but as I said, also a high-level association area, and from here to the highest association area, the prefrontal cortex, the area around the principal sulcus, where information is fed into the prefrontal cortex. So we go in with single cell electrodes, usually many of them at the same time, and record single cell activity from the neurons there. And today I will focus on the prefrontal cortex and not talk about the posterior parietal cortex. And indeed, when we do this, and this was very exciting for us, and probably also one of the reasons why I'm talking here, is we find neurons that are tuned to the number of dots that the monkey is seeing, but not only seeing, but also memorizing. So we are finding a working memory correlate for the monkey memorizing number. Here is one such neuron. This is again a dot raster histogram, as I explained before. This is a smooth peristimulus time histogram that we call the spike density histogram, but it shows basically the same when the functions are going up. That means that the neuron is increasing its firing rate. In this case, we were presenting numerosities from 1 to 30, so really a broad range. And indeed, we found neurons that were specifically tuned to a specific number that the monkey was encoding and memorizing. This neuron is a neuron that we say is tuned to numerosity 2 or has a preferred numerosity 2. That's the dark blue curve that you see here. And this is during the presentation of the stimuli. So this is the visual impression of the monkey, if you like. And this is during the working memory delay. And you see both during the encoding stage and the working memory stage, this neuron has an increased and a significantly increased firing rate for numerosity 2. We test this with ANOVAs, of course, so that to make sure that this is not an accident. And you see that the other numerosities are encoded less, and they progressively and systematically drop off relative to the preferred number 2. So here, for instance, is 4, and the black curve is for 1. When we integrate this information and plot it as standard tuning functions, we get beautiful tuning curves. This is the tuning curve during the sample presentation. This is the tuning curve of the same neuron during the working memory delay period. And you see in both cases, we have the preferred numerosity 2, which is the highest discharge of this neuron. We also find neurons that are tuned to any other numerosity that we are presenting to the monkey. Here is just one more example neuron that was beautifully tuned to numerosity 6. So you see the tuning function is peaking for 6, again, both during the sample phase and during the working memory delay period. I should say that many of the neurons have the same preferred numerosity in the sample and the working memory delay, but not all of them. Some of the neurons can be totally ignorant about the visual presentation of the numerosity, but then start to highly become activated during the working memory period. So there's a mixture of different temporal profiles of these neurons. We find these neurons, particularly those that are interested in working memory activity, primarily in the prefrontal cortex, where in some monkeys we have up to a 30% of randomly collected neurons that are significantly tuned to a specific numerosity, particularly during the working memory period, as I said. This would also be reflected in the posterior parietal cortex, of which we think is the input stage of the system, the first stage in the brain where this kind of information is represented. And one reason, of course, why we work with non-human primate is because we want to compare it to what's happening in our human brain. And from comparative reason, I brought here a functional imaging meta-analysis where about 30 fMRI studies have been combined and superimposed, if you like. And whenever we humans, and this is now a human brain, left hemisphere, slightly graphically inflated so that we can look into the sulci, whenever we humans are performing, calculating, memorizing numbers, these two areas, the prefrontal cortex and the posterior parietal cortex, are becoming activated, and this is really emphasizing. I'm strongly convinced that what we see in the monkey brain is an evolutionary precursor of our own number competences. The neurons, as I said, all have their preferred numerosity, and when we normalize them and average them according to the preferred numerosity, and in this case, this was a study where we presented only numerosities from 1 to 5, then we end up with these average tuning functions for different preferred numerosities. So for instance, the cyan blue curve here, this would be all the neurons that are tuned to numerosity 2 during the delay period, during the working memory delay period. And we see classical tuning functions that are also highlighting numerical distance effects. So the psychophysical finding that it is easier for us or for the monkey to discriminate numerosities that are very close. So for instance, it's simple to discriminate 5 from 2, and you see a huge decrease in activity compared to discriminating 2 from 3, which is much nearer, where the neuron has problems to differentiate these two types of information. You might also see, if you analyze these data more closely, that the tuning functions are becoming broader with an increase of the absolute numerosity, and this is a sign of the so-called numerical size effect, indicating that at a given numerical distance, it's becoming progressively harder to estimate the difference between two numerosities. And this is what the monkeys also show in their behavior and what the neurons show. So we have evidence that the neuronal activity is really a correlate for the monkey's behavior. But we want to have a better correlate, and in this case, we resort to what we call error trials. Now and again, the monkeys are making errors, and they're mixing up the numerosities. And these error trials are, for us, very informative in terms of understanding what the neurons are doing and whether they are behaviorally relevant. Because if you think about it, if the monkey is mixing up or cannot memorize the proper numerosity, you would expect the neuron to show a different discharge compared to when the monkey is doing a correct trial. And this is in fact what we find. This plotting here is basically all these plots, again averaged according to the numerical distance from the preferred numerosity. So you put each of these functions, superimpose them according to the preferred numerosity, and the plot you get is what you see here, a nicely peaked function. And the question is, what are these same neurons doing when the monkey is making an error? And the red curve here shows you the error trials. And you clearly see that the peak of this tuning function is basically clipped. In other words, when the animal is forgetting about the numerosity it was seeing, the neuron is not, it correlates with the neuron not being able to encode its preferred numerosity properly. And if the neurons are not encoding their preferred numerosity, the monkey is prone to making an error. This is the message here. Again, emphasizing that this is behaviorally relevant. Now we move on to something quite more complex, at least for a monkey. This is the question, can the monkey follow certain rules or switch between rules in a working memory delay period? So here, this is a different kind of task that I briefly want to explain. You see the monkey again facing a computer monitor, fixating, and then it's presented with a sample numerosity, 5 dots in this case. The monkey has to memorize it during the first delay period, but then comes a rule cue, in this case, the color red. And the color red, the monkey learned in previous trials, is associated with a greater than rule. So whenever the monkey sees the greater than rule, it is supposed to pick the numerosity that is greater than the one it had seen in the sample stimulus. And after a second delay period, and we call this the rule delay period, the monkey is presented either with more numerosities, with more dots, 8 in this case, and the monkey is responding by releasing. It's following the greater than rule because 8 is greater than 5. If the monkey is seeing only 3 dots, then the monkey is not allowed to respond because 3 is smaller than 5, and the monkey is supposed to follow the greater than rule. In the other half of the trials, obviously, we had the monkey cued for the smaller than, for the less than rule, and this was done by a blue circle. So whenever the monkey saw a blue circle, it had to respond to fewer dots compared to what it had seen in the sample period. Behaviorally, we would be ready to go, but from a physiological point of view, we have now one confounding factor, which is exactly the color, because the color corresponds exactly with this abstract rule. So if a neuron would respond to the rule, we would not be certain whether it's really to the rule or to the color of the rule cue. So the poor monkeys had to learn a second set of rule cues that were without a color. And the second set of rule cues were a drop of water with a white circle that also indicated the greater than rule, and the less than rule was indicated by a white circle without a drop of water. So we had 4 conditions with 2 different rules being cued. And this allowed us then to take the color of the rule cue into our multifactorial ANOVA design and making sure that the neurons are not responding to color, but if they responded really to the abstract rule cue. The monkey was not only doing this task in relation to 5, but it was facing 2, 3, 5, 8, and 13. And all of these different numerosities, for all of these different numerosities, it had to apply the greater than less than rule. These are the performances of the monkeys. The cool colors are the less than rule cued in the 2 different cool cues that I just explained. The warm colors represent the greater than rule, again cued in the 2 different rule cues. And here you see the percentage correct plotted against the sample numerosities. And I hope you see both monkeys are doing very nicely. They are between 80 and 90% on average correct. So they really understand the rule and they can follow this very abstract numerical rule. When we recorded from the prefrontal cortex, and here you see now a right hemisphere from a monkey brain, and this red circle is basically where we had our recording chamber and we were recording from, we found beautifully abstract responding neurons that were significantly encoding which rule the monkey is going to apply as soon as it was facing the comparison stimulus. We call these rule selective neurons, and they are depicted here. This is again a spike density histogram. For us now, the most interesting phase is the second delay period after the rule had been shown, but very importantly, before the comparison stimulus was shown. And that's very important for me to emphasize. The activity that we see in this delay 2 period cannot be a motor preparatory activity because the monkey does not yet know if or how to respond because it's still lacking the comparison stimulus. So the activity we see in this delay 2 period is a very abstract signature of the monkey loading a rule without yet knowing what to do with it, a very abstract type of signal. And indeed, we find about 20% of the neurons are rule selective neurons. And here you see again, based on the cool colors representing the less than rule and the warm colors representing the greater than rule, that this neuron towards the end of the delay 2 period is nicely increasing and significantly increasing. Again, we tested this with our statistics. Its firing rate whenever the monkey is about to follow the less than rule, but before it is really executing it. And this neuron doesn't care about the 2 different rule cues. As you see, the 2 different rule cues activities are basically overlapping. All it cares about is the rule that the monkey is following. So this is a rule selective neuron for less than, and we, in equal proportions, we also found neurons that were rule selective for the greater than rule. So here at the end of the delay 2 period, this neuron systematically increased its firing rate when the monkey chose the greater than rule and was not interested in the less than rule. And here you see a magnification of these kinds of activities. We found, as I said, about 20% of the neurons that were rule selective, about 10% were selective to the greater than rule. And in the surface plot, they are now encoded in the color red. And the other 10% were selective for the less than rule. Each of these neurons showed a specific temporal trajectory. So they all together started to increasingly encode the greater than rule and the less than rule while the monkey was approaching the end of the delay 2 period, which again is a very classic working memory period. But now it's not only about memorizing numerosity, but it's really processing the scene's sample numerosity in relation to the rule that the monkey has to follow. We now and again are also interested in how these neurons change the coding properties as a function of neuromodulators. And the neuromodulator that is for us most interesting is dopamine. Dopamine is particularly, dopamine neurons from the midbrain are particularly projecting to the prefrontal cortex. And it is known from the clinic that whenever neuropsychiatric diseases occur, an imbalance of dopamine is involved. However, hardly anyone knows why this imbalance is happening and what it is doing to the neurons. So in this kind of projects here, we are trying to make sense of how the neurons are affected by dopamine. And we do this by iontophoresis recordings. So we have in-house built electrodes. At the tip of this electrode in the center, you see our metal recording electrode with which we can record single cell activity. And this electrode is accompanied by 2 flanking barrels that are open at the end. And these barrels are filled with certain drugs that we can eject by applying very mild currents. So these drugs are charged, and by applying subphysiological currents, we can eject these drugs in very miniature amounts and see how they are affecting the neurons that we are recording with this electrode tip here. And that's this electrode that we have to build. The dopamine system comes in 2 different shades. So it is known that we have 2 different, as they're called, dopamine receptor families, the so-called D1 receptor families and the D2 receptor families. They function differently and they have different channel kinetics and so on and so forth. I'm not going into the details, but with the drugs we have available, we can target each of these dopamine receptor families individually. And we do this, for instance, by applying this drug SKF, that is a D1 family agonist. Agonist means that it is activating these types of channels. And we can also, in different trials, use quinpirole as a D2 agonist. Again, agonist meaning here that it's activating or exciting these receptors on the membranes of these neurons from which we are recording from. And the protocol that we are using then goes like this. So first we record these neurons in a control situation without any drug. Then we switch over to ejecting the drug, record the same neurons, and then if we are lucky, we can go back and forth for as long as we can keep the neuron at the electrode tip. What we find in the very same protocol that I just alluded to in this rule switching protocol is that the neurons, the rule selective neurons, are highly affected by the dopamine influence. We see, for instance, this is a single cell that is not rule selective during the control condition, but when we stimulate the D1 receptors, it all of a sudden becomes beautifully tuned to the less than rule. So we are, if you like, creating a rule selective neuron by stimulating dopamine 1 receptors. This is a single neuron, and I should also say this is during the delay 2 period, so after the rule cue has been presented, and we find such 20 of these neurons, and these are the normalized average responses, firing rate responses of these neurons. And you see very clearly whenever we apply or whenever we stimulate dopamine 1 receptors, the neurons on average are becoming more selective to their preferred rule. And we also statistically verified this. So obviously, D1 receptor stimulation is enhancing rule selectivity in these neurons. For other neurons, we had a D2 stimulation with quinpirole. Again, a single cell example, population average firing rates, and also for D2 receptor stimulation, we found a significant increase in rule cell activity for these neurons, and this was also highly significant. Now, on the one hand, we were happy to see that dopamine had an effect on our neurons. On the other hand, we were a little disappointed because it seemed as if both dopamine families had the same effect. They were enhancing the rule coding properties of our neurons. However, when we looked more closely, we found a nice difference that is pointing to mechanistic difference of these two dopamine receptor families, because what is happening for the D1 stimulation is that the preferred rule is encoded better, whereas the non-preferred rule stays about at the same activation. And this looks very different when we are activating the D2 receptor family. In this case, the preferred rule stays constant, but the non-preferred rule is being suppressed. The consequences is the same. Rule coding is increasing, but the mechanism here is a very different one. And we were very happy to see this. So summing all of this up, the dopamine 1 receptors are modulating or increasing the preferred rule. The dopamine 2 receptors are increasing the encoding of the non-preferred rule. And that's the difference between these two receptor families. Now, in the second part of my talk, I switch over to our second beloved animal model, and these are the carrion crows. Carrion crows are birds, as I'm sure all of you know. And birds actually originated from dinosaurs, and dinosaurs are reptiles. So some zoologists say that birds are flying reptiles or feathered reptiles, and this is not entirely incorrect, but it's also a little weird. What you see here is a very simplified tree of vertebrate life, how different classes of vertebrates emerged over time. And for me, the most important branching point is here. This is about, actually, according to most recent fossil founds, I need to update this, 360 million years back. This is almost 6 times longer than the extinction of the dinosaurs. At this point, the last common ancestor between us mammals and reptiles and birds on the other side lived. Since this time, this evolutionary eternity, these two animal classes have nothing to do with one another. And you can imagine there's a lot of time to develop brain areas differently. And this is exactly what happened. When we look at the pallium or the telencephalon in a very primordial or embryonic stage, then and this is what you see here. So this would be a very embryonic stage with 5 brain vesicles. The one that is most important for us is the telencephalon, the most anterior one. Telencephalon comes in 2 hemispheres. And when we make a frontal section through one of the hemispheres, we end up with a picture that you see like here. The telencephalon contains 2 areas, the subpallium, that is not interesting for me, that is turning into corpus callosum, not corpus callosum, striatum and other parts of the brain. The colorful parts here is the so-called pallium, the roof of the telencephalon, and this is where the magic is happening. People are discriminating 4 different territories of the pallium. A medial part that is turning into the hippocampus, a dorsal part that is turning into the cerebral cortex in our mammalian brain, the lateral pallium that is paleocortex and is mainly olfactory, and the ventral pallium here in green that in birds is becoming the major integration center, whereas in our brain it is part of the claustrum and other relatively small areas in the brain. So when we follow the trajectory, the developmental trajectory of these different areas, again, here we see in a primate brain, in a mammalian brain, the cerebral cortex, the neocortex, is all blue because it stems from the dorsal pallium, whereas in the bird brain, the major integration centers are all green because they come from the ventral pallium. So on the one hand, we have a deep homology, it's all pallial. On the other hand, we have also convergent evolution because different territories of the pallium turn into the major integration centers in these two different species of animals. And another very important aspect is that the entire microcircuitry in the pallium is very different. In the mammalian cortex, it's well known that we have a basically 6-layered nice cortex with certain repeated circuitries for thalamic input through the cortical layers and then the output to subpallium or brainstem areas. It's all happening across these layers, but the bird brain does not have a neocortex proper. It has more a nuclear arrangement of all its sub-areas, and here we see the microcircuitries mainly represented across these nuclei in the pallium. And this is what it's making this brain of the birds very interesting for me as a comparative biologist, because now we have a chance to investigate what are the general physiological properties for any smart animal, because corvids among the birds are extremely smart, as are primates, as we know. But the anatomy is very different. And in case you ask whether corvids are in any way special among the birds, this is one of my answers. The answer is clearly yes. You see here the top view of crow brain. Bottom would be anterior, up is posterior. This is the cerebellum, and these are the huge, very hypertrophied telencephalic hemispheres of a crow. And you compare it to the same view of a pigeon brain, and you immediately realize that the telencephalon is only about half as big. And this is meaningful because in addition, you need to know that both the crow and the pigeon have about the same body weight, so about half a kilogram. So this difference cannot be explained simply based on different body weights. It means basically that crows and corvids in general are extremely smart because they have such a high number of neurons in their brains. And we also know by now that the density of the neurons is about 2 to 3 times higher compared to a primate brain. So birds really packed as many neurons into the same volume of brain tissue, or 3 times more neurons into the same volume of brain tissue compared to even a primate. Why did they do this? Well, the answer is probably because they still need to be able to fly. So evolution did everything to make these birds as lightweight as possible. And the way to do is to pack as many neurons in the same volume as you can possibly do. Now, we have our crows that we are breeding here in Tübingen, and we hand raised them to have them as tame as possible and that they're also interacting with us. And we train them on different tasks. Here, this is one of the tasks, which is exactly as the one I showed you before for the monkeys. It's a delayed match to numerosity task. The only difference here is that the crows, because they do not have hands, they are not grabbing a manipulandum and release it to respond, but they peck at the screen in order to tell us that they have seen the same numerosity now twice in a row, and they are not allowed to peck at the screen if they are seeing a non-match. And I would like to show you this movie here with one of our crows. So the crow is facing this computer monitor where the stimuli are going to appear, and you see the same task that I just indicated, this delayed match to numerosity task. Because this is all operant conditioning, we of course need to reward the animals. So here at the bottom you see a feeder, and whenever the crow is doing something correct, it is allowed to get a choosing mealworm as a reward. This was a relatively early training stage. I should mention to you, when we are recording, the animals are only allowed to peck at the test stimuli. So they have to keep still while we are presenting all the other stimuli and during the delay period. In this case, you will see that the animal is also pecking at the sample stimulus. But what it is not allowed to do is to peck at the non-matched stimulus. So please enjoy this video. So the sample is 1, delay, and 1 would be the match. This was a correct trial and it's rewarded. Next trial, 3, 2 is a non-match, and 3 is the match. So again, you see that the animal in the delay period has to memorize the numerosities it had seen in order for it to choose the matching numerosity as soon as it appears. We are now going into record from the brain. The only difference will be that we are implanting under general anesthesia electrodes that are chronically implanted, attached to microdrives, and that there would be a preamplifier attached to the bird and a cable dangling from the head while the animal is working, but otherwise it would look exactly the same. We are now recording from an area that is supposed to be the equivalent of the prefrontal cortex of the primate brain. It's depicted here on this collage. It's the so-called nidopallium caudolaterale, or in short, NCL. It's a very cumbersome name, a very anatomical name, but I cannot help it. This nidopallium caudolaterale evolved totally independent from neocortex or from the prefrontal cortex in birds, and that's what's making it very interesting for us. And funnily, it's located at the back of the telencephalon, not at the anterior part as in our brain. So that's one of the prime differences. It receives very heavy dopaminergic input from the midbrain, just as the prefrontal cortex. It receives highly processed sensory information from secondary visual, auditory, somatosensory areas, just as the prefrontal cortex. These are these orange lines here. It has connections to the hippocampus. It receives input from the hippocampus, just as the prefrontal cortex. It has output to premotor cortex areas, and it has reciprocal connections to limbic structures, such as the bird equivalent of the amygdala. In other words, all the connections, and we verified this 2 years ago by track tracing studies in our crows, all these connections indicate the nidopallium caudolaterale as a high-level association area in the brain of these birds. And this is what we are looking for, because after all, when we talk about working memory, we need to have a high-level association area that is able to memorize and process all sorts of information and output it to motor areas after that. So we are now going in and record from this area, the nidopallium caudolaterale, this green part of the brain. And so here you see this depiction. And indeed, we again found neurons that were beautifully tuned to numerosity, in this case, during the sample period, to numerosity 20 in this case. And here is a neuron that's more important for the talk currently, that is very nicely tuned and shows this ramping activity during the memory delay, when the crow is memorizing its preferred numerosity, which in this case for this neuron is the preferred numerosity 2. You see clearly the orange function goes up, and this is significantly different from all the rest of the activity. So again, here in a crow, just as in the prefrontal cortex of monkey, we find neurons that are significantly tuned to a specific feature, the numerosity, during the working memory period. And I don't have the data with me, but we also tested some of these neurons during error trials when the crows made mistake. And we saw exactly the same as in the monkeys, that the neurons were not properly encoding their preferred numerosity, causing the crow to be prone to make mistakes. In the second part of my crow talk now, I'm again switching over to a much more complex task, which is again a rule-following task. This time, the animal is not following a greater than less than rule, but another abstract rule, which is a same different rule. And I briefly explain what the animal has to do. So again, it's sitting in front of our touchscreen monitor, and it is presented with a sample image. This is this kind of orange image that you see here. The crow has to memorize the sample image during a first delay period and a second delay period, and its job is afterwards to peck at either the same or a different image. In this case, the animal was cued for the same rule. So the blue circle meant whenever you see the same picture again, peck at it, choose it. Do not choose the picture that looks different. So in this case, the orange one would be pecked at. Of course, we also had the corresponding rule, the different rule, indicated by a red circle. Whenever the crow saw a red circle, it was informed that now it should peck and choose the image that was different from the sample, different from the one it remembered. And I think it's immediately clear this is a task that the animal can only do if it is A, remembering the sample, and B, following the rule, whether it should choose the same or the different image. And so again, here we had the problem of the color as a potential confounding factor. And in this case, we had an even better alternative rule, which was now in the auditory domain. So whenever the animal heard a deep frequency sound, this was the rule cue also for pick the same image as the one you have seen before. And if it heard a high frequency tone, then it was informed that now it should peck at the different image. And we switch between these rules randomly. So on any single trial, the animal was not aware before the trial which rule was in effect. They beautifully nailed this task. And for an animal, I can tell you it's a pretty complex task. It requires a lot of training to make sure that they can do it. Here are the correct responses of two crows, D and P here. The black column are our so-called baseline trials, where the animals were working with images that they knew already from previous trials. And in order for us to really exclude that the animals were memorizing anything that we didn't want them to memorize, such as the identity of the images, in the trials that are depicted here in green, they saw completely new images on every single trial and only once. And you see very nicely, they do not care about whether they see familiar or novel images. All they care about is the rule and that they can follow the same and different rule. And they have a performance of 90%, which by all standards is really excellent for any animal. This is a movie where you see one of our crows working. It's all happening relatively quickly. Again, I explain the first one or two trials, and then I hope you can enjoy the rest of it. That's the ready cue. So this is the sample, this image. Red means pick different, and then it pecked at a different stimulus. This is the next trial. Sample, different rule, and it pecked again at a different image from the two options. That's the same rule cue, and it pecked at the same rule, same cue. So whenever we make these movies, they're a little nervous because we are opening their box, and they have a tendency to look outside because they're not used to work with an open door. But you see they can do this very concentrated and very focused. Now, of course, we wanted to know, can we find a neuronal correlate of this rule switching in the working memory period? And again, the answer is, of course, yes. Otherwise, I wouldn't talk about it. We recorded from the nidopallium caudolateral neurons again while the crows were performing this task. And here is one almost unbelievably beautiful rule-selective neuron that we found. This is the top. You see again the trial layout with rules, either the same or different rules, the delay 2 period and the choice situation. And again, I'm mostly interested in what's happening after the rule has been presented in the delay 2 period, because this is the first period where the animal knows which rule to follow. But again, it cannot prepare any motor act because it first has to see the choice stimuli in order to know which one to pick. And dot raster histogram, spike density histogram, the red color stands for the different rule, blue color stands for the same as sample rule. And you see this neuron is dramatically and exclusively increasing its firing rate whenever the animal is following the different rule. If you have a neuron like this and you hear while the animal is working, you can predict which rule the animal is following online while it is working. That's how beautiful and selective this neuron is. And you see in all the other trial phases, the neuron didn't care at all about anything else. It only cared about the different rule. And you see this is a huge increase in the firing rate. Again, we wanted to know whether this activity is behaviorally relevant. It might just be an epiphenomenon. So we looked at the error trials. And when you looked at the error trials and you think about it, an error trial means that we cue the animal for the same rule, but it was following the different rule. So in this case, you would expect that for the same rule, you would find a similarly high activity as for the different rule because the animal was following the different rule. And this is in fact what we saw. So this blue curve here, this is now cueing the same rule, but the animal was erroneously following the different rule. And you see a huge increase in firing rate that is quite comparable to what the animal is doing in correct trials and very different to what the animal is doing when it is actually following the same rule. And when we cued the different rule and the animal was following the same rule, then we found a very decreased or almost no activity, as we would have expected. And that clearly indicates that these neurons are behaviorally relevant for the animal to follow this abstract rule. We verified this with a classifier approach. So we trained the classifier with 50% of the recordings so that it could learn about the neuronal discharges when the animals made correct choices. And then we tested it with the other 50% of the recordings that the classifier had not seen so far, and asked the classifier, can you predict, simply based on the neuronal recordings, whether the animal is following correctly the cued rule or not? And the black curve here, these are all the correct trials, and you see the classifier can predict with almost 100% certainty, based on this population of neurons, whether the animal would follow the correct rule. And also during error trials, we see that towards the end of this delay period, the classifier is switching over to the other rule that was not cued, as we would have expected during error trials. So again, clearly indicating that this is relevant for the animal's behavior. Now, during the last part of my talk, I briefly want to kind of summarize and provide an outlook about how many people, many physiologists currently are envisioning the code for working memory in the brain. What I was talking about now throughout of my talk mainly was what we call the sustained or the static code. The static code meaning it's based on so-called sustained activity of neurons or persistent activity of neurons. And that means that a neuron, as soon as it likes a specific feature of the stimulus, a specific memorandum, it is increasing its firing rate during the delay period. So imagine this is our working memory delay period from the beginning to the end of this dotted line. It's increasing its firing rate and it maintains this high level of firing rate up until the end of the delay period. So this would be sustained activity, giving rise to a static code. Static because the neurons are encoding the same preference across the entire working memory interval. if you have a neuron like this, you can imagine that if you train a classifier at the beginning of a delay period, and the classifier is extracting this kind of information from this neuron, it should also be able to do a high-level prediction in the middle and then at the end of the working memory period. Why? Because the neuron is not changing its selectivity. And if you do this, you would end up in what we call a cross-temporal classification. So the timeline of the training the classifier would be at the bottom at the x-axis, and the timeline of testing the classifier would be on the y-axis. Again, if you train the classifier at the beginning of a delay period, the classifier would perform exactly as well at the end of the delay period, and this would give rise to such a very characteristic high percentage classification rate of such a neuron. In recent years, however, a second working memory code is discussed, or there's evidence actually for this working memory code, which is called a dynamic working memory code. And it is based on so-called dynamic or transiently discharging neurons, as you can see depicted here. So the difference compared to the sustained cells is that these neurons are only briefly selective during the delay period. And we have this neuron to have selectivities at different time points throughout the delay period, almost like a relay, as a neuronal relay, if you like. And if you have such a neuron that is only responding to a certain feature at the beginning of the delay period, a classifier would not be able to predict what is happening, what feature the animal is following or memorizing at the end of the trial, because this neuron would not discharge and not respond. And what you find then classically is only at the so-called diagonal, such a confusion matrix, a high-level classification performance. Now, the last question I'd like to address, and then I stop, is how can we envision a sustained neuron, a neuron that is continuously firing throughout a working memory period for many seconds on end, even though the stimulus itself has long ceased. This is not a trivial problem, because when you think about it, during the working memory, there is no stimulus that could provide an energy for a neuron to become active. So this kind of activity, the sustained activity, this ongoing activity, somehow has to be generated within the brain and within the neuronal network. And there are two basic textbook explanations of how this could happen, two mechanisms. One is simply based on one single cell alone, based on the intrinsic properties of the neuronal membranes. I'll come to this in a second. And the second mechanism is based on larger networks of neurons based on recurrent synaptic connectivity. And I briefly try to elucidate what I mean by that. So the first mechanism, intrinsic membrane properties that would generate persistent activity. This is actually something you can see in intracellular or patch clamp recordings. Actually, intracellular recording. This is the timeline again. Here would be the action potentials of a single neuron. And here you see a stimulation pulse. So current is injected in order to excite the neuron. And if you do this long enough, you get such a neuron into a permanent firing rate that it keeps holding for many, many seconds. And this is a single neuron, so it has to have to do with membrane properties. The idea is depicted here. So with excitation, you are opening or you're depolarizing the membrane, and the idea is that voltage-gated calcium channels are now becoming activated and open. Calcium is flowing in, and this increase in calcium is continuing to depolarize our membrane, so that non-selective cation channels are being opened a little later that are generating an action potential. And this depolarization itself, and in turn, is activating again our voltage-gated calcium channels. So you have a constant recurrent influx of calcium and cations in order to keep this membrane constantly depolarized so that it's generating firing rates, action potentials on end as long as the refractory period is not exploited. The second type of mechanism many people think about, including we ourselves when we are recording from the prefrontal cortex, has to do with recurrent synaptic connectivity between larger networks of neurons. And this comes in three different shades that are depicted here. So one would be that we have persistent reverberatory network activity, activity between widely separated populations of neurons depicted here, such as the neurons in the classical association areas of the prefrontal, the posterior parietal, and the temporal lobes. They are all reciprocally connected, as we know based on anatomical studies. So an activation in one of these areas could trigger the activation in the other. And if they are being connected, they could re-excite each other basically forever. Of course, not forever, because at some point the stimulus would stop this entire reverberation. The second, more small-scale idea is that we have to do with recurrent excitatory connections within active populations of neurons that operate locally, for instance, within the prefrontal cortex. The idea that we have glutamatergic excitatory pyramidal cells that are connected with one another based on axon collaterals and based on these delay times from one neuron to another one, they could constantly reactivate each other and maintain this activity like an echo, basically, in this entire network. There are many computational models about this idea. And the last, even more fine-grained activity is that we have mutual reciprocal inhibition networks between two populations of neurons that you see here that then basically work in type of like an oscillator. When one element is excited, the other one is inhibited and vice versa. So we have a mechanism of positive feedback that is termed disinhibition. All of these three ideas would allow neurons to fire continuously, even though there is no new input to the network. And this is the end of my talk. I briefly summarized, so I try to define what is working memory. Again, it's not just any short-term memory, it's an active, a conscious, distractible, capacity-limited type of memory. We find the prefrontal cortex in primates and the nidopallium caudolaterale in crows as the core hubs for working memory activity. I showed you that neurons in the monkey and in the crow are nicely activated during very classical working memory periods in delayed match to sample tasks, but also in more complex rule switching tasks. And finally, I tried to sum up with the ideas that we have a static code and a dynamic code that is interacting in these cortical areas. So it's probably not only a static code or only a dynamic code, but the evidence speaks for both of these codes being combined to certain extents in these areas. And yes, with that, I thank you very much for your attention, and I stop here, and I would be happy to take some of your questions.